Metabolism of diacylglycerol. Diacylglycerol is a glyceride consisting of two fatty acid chains covalently bonded to a glycerol molecule through ester linkages. The combination of these two fatty acid chains may also be very different. For example, the acid chain, which belongs to the first carbon, should always be saturated. However, the fatty acid chain belonging to the second carbon should always be unsaturated. And in most of the cases, it is polyunsaturated. Also, we should not forget that the identical molecular formula doesn't mean that the structure of compounds is the same. So there are two types of isomerisms for diacylglycerol. The first one is constitutional, or it is often called structural isomerism, which means that there are differentially linked atoms or functional groups in different isomers of diacylglycerol, but in this case they will be called regiomers. So there are three types of them: REC13, SN12, and SN23. And the second type of isomerism is spatial or stereoisomerism, which means that a different geometric position in space, that functional groups form different geometric positions in space. So there are two types of isomers, or they are often called enantiomers. It is SN12 and SN23. There are a few ways of synthesis of diacylglycerol and its isomers. The basic one is the reaction which is catalyzed by lipase, and we get 3 isomers of diacylglycerol from triacylglycerol. So the second one is the reaction which is catalyzed by either phospholipase or transferase from phospholipids of cell membrane. But here we get only one isomer, SN12. And the main reaction, which will interest us in the next slides, is the third reaction, which is catalyzed by phosphatase, and we get SN12 isomer from phosphatidic acid. It is the main reaction. It is the key, the signaling way of diacylglycerol. Also, there is the fourth reaction, which is catalyzed by acyltransferase. From monoacylglycerol we get 3 isomers of diacylglycerol. But this reaction is very seldom, and the only 3 ones, the first 3 ones, are the main ones, so that's it. The utilization of diacylglycerol is closely connected with its ways of synthesis, and the diacylglycerol which we got from triacylglycerol, monoacylglycerol, phosphatidic acid, and phospholipids of cell membrane, it can be transformed into these compounds again, but by different enzymes. So we get monoacylglycerol, phosphatidic acid, and triacylglycerol from every type of diacylglycerol isomer. So now let's talk about diacylglycerol as the secondary messenger. So the diacylglycerol isomer, which will be the secondary messenger in the cell membrane, is gotten from phosphatidic acid. So from phosphatidic acid we get the CDF diacylglycerol. CDF is cytosine monophosphate. After that we get CDF diacylglycerol, and the next step is transferring the inosite to this compound, and we get phosphatidyl inosite. After that come two reactions of phosphorylation, and we get, after these two reactions, phosphatidyl inosite 4,5-biphosphate, or it is often called diphosphate. But how does our diacylglycerol appear? The one that will be used in signaling way. So the phosphatidyl inositol 4,5-biphosphate is transformed into two compounds. One of them is inositol 3-phosphate, and the second one is diacylglycerol. And this reaction is catalyzed by phospholipase C. After that, our diacylglycerol is transformed by DAG lipase either into monoacylglycerol or arachidonic acid. And arachidonic acid, as you know, can be transformed into prostaglandins or leukotrienes. Well, it depends on the enzyme. So now, the signaling function. For example, there is an agonist which interacts with the receptor. After this reaction activates G proteins, to be specific, GQ proteins, and the subunit, the alpha subunit of GQ protein, is the enzyme GTFase. So it uses GTF to get energy and to activate the phospholipase C, which is also located in plasma membrane. After it is activated, the phospholipase C is reacting with phosphatidyl inositol 4,5-biphosphate in plasma membrane, and we get two compounds out of this compound. Our two compounds are, as I told you earlier, inositol phosphate or inositol 3-phosphate and diacylglycerol. Diacylglycerol, if we talk about the classic protein kinase C, together with ions of calcium, activate the protein kinase C enzyme, which is the enzyme that causes substrate phosphorylation, and it is used in cell differentiation and mitosis. This type of signaling is a mechanism for such hormones as vasopressin through V1 receptors, adrenaline through alpha-1 adrenoreceptors, and angiotensin II. So the V1 receptors are located in smooth muscles, in arteries and veins, and also in hepatocytes. And the effects which they cause are vasoconstriction, platelet aggregation, and glycogenolysis. The alpha-1 adrenin receptors are located mostly in arterioles, so the effects are the constriction of arterioles, which results in rise in arterial pressure. And for AT1, these receptors are located in smooth muscles, in arteries and veins, also in adrenal cortex and proximal tubule. And the effects are vasoconstriction, the production of noradrenaline, and aldosterone. Let's talk about the protein kinase C. This enzyme has two domains. One is regulatory and the second one is catalytic. And you should also remember that the inactive form of this enzyme is located in cytoplasm and the active form is membrane-based. So when protein kinase C is activated by diacylglycerol or calcium ions or other compounds, it is moved from cytoplasm into membrane. Also, there are three isoforms known for protein kinase C, and as you see, three types of compounds are used to activate classic protein kinase C, which has all domains, and the compounds are diacylglycerol and calcium ions. Also, instead of diacylglycerol, protein kinase C also can be activated by furbol esters. Also, there is a new isoform of PKC which doesn't have the C2 domain, thus is not activated by calcium ions, so you only need DAG or furbol esters. Also, there is an atypical one which doesn't have C2 and C1 domains, so it can't be activated by DAG, calcium, or furbol esters, but it is activated by phosphatidylserine. Also, there is a model which is called the three-pool model of DAG metabolism. So, according to this model, there are three pools of DAG in the cell. So, the first one is located in endoplasmic reticulum or Golgi apparatus. These diacylglycerol isomers SN12 and SN123 are only used to get triacylglycerol and other compounds. The second pool is located in lipid droplets, and it is REC13 and SN23 isomers. So isomers from this pool are also used only in getting other compounds. And the third pool, which is located in plasma membrane, is the main pool for getting the diacylglycerol for a signaling way, because we know that for signaling or for diacylglycerol to be a secondary messenger, it has to be located in plasma membrane. So when it is located in cytoplasm, it doesn't act as the catalyst for protein kinase C, which is also located in cell membrane. And that's why the main concentration for signaling is in plasma membrane. Also, we talked about the furbol esters. They are one of the strongest activators of PKC, and their effect is the same as for DAG, but it is much longer. For example, the DAG can affect a protein kinase C for about seconds or minutes, whereas for furball esters the approximate amount of time is hours or even seldom days of activating protein kinase C. And during this period of time, furball esters constantly interact with PKC, so the latter produces substances that intervene in normal cell regulation processes, such as differentiation and mitosis, which make them potential factors for carcinogenesis. They are also known as activators of respiratory burst and macrophages. Also, there are some references which were used in making this presentation. Thank you very much.